Hi, I am Marion Bornhorst, and today's date is August 27, 2019. And I will be talking about case connections for Unit 2, Uni Biochemistry. And I've chosen to go over case connection 2 and case connection 3. So we'll begin with case connection 2. And case 2 presents us with a topic based on how glucose uptake is regulated by the cell in different tissues. And in this case, we focus on a glioblastoma tumor in the brain that is depicted by both MRI and PET scan. And the case wants us to determine how or if the glucose uptake differs in a tumor as compared to other tissue types. However, it is important to begin by reviewing how exactly other tissues take up glucose and how that process is regulated before beginning that comparison. So the major tissues that we'll be discussing include the liver, skeletal muscle, brain, and adipose tissue. And all of these tissues utilize glucose in the fed state to provide energy for underlying metabolic processes. However, the way in which glucose enters the cells of each tissue type vary. So to begin, adipose and skeletal muscle tissue, they uptake glucose through a primary glucose transporter that is termed GLUT4. And this glucose transporter is controlled primarily by the hormone insulin, which is secreted by the beta isolate cells of the pancreas in response to an increased concentration of glucose in the portal blood. So GLUT4, unlike the other primary glucose transporters, is intracellular, and an early response to insulin migrates the GLUT4 vesicles to the cell surface, where they can fuse with the plasma membrane, exposing active glucose transporters and then allowing for glucose uptake in those tissues. So this system of using GLUT4 allows for glucose uptake to be regulated by insulin, and also in fasting states, the skeletal muscle and adipose tissue are taking up less glucose because there is also less insulin. So focusing in on the liver, it takes up glucose in an insulin-independent manner, so it uses a glucose transporter, GLUT2, instead. And this transporter is always present on the cell membrane. Insulin, however, is thought to indirectly promote uptake in the liver by encouraging the synthesis and storage of glucose and glycogen, but it is not directly affected by insulin in any way. So the last tissue that I will focus on is the brain, which is also a tissue primarily found in this case study. Just as the liver, the brain takes up glucose in an insulin-independent manner, and in this case the brain uses GLUT transporters, GLUT1, and then also GLUT3 to transport glucose into the cells. So GLUT1 specifically transports glucose across the blood-brain barrier, and GLUT3 controls the uptake of glucose into the neuron. So overall, with brain metabolism, the brain will always preferentially oxidize glucose, except in the case of extreme starvation, under which the brain could also employ the use of ketone bodies for fuel that are released by the liver in that case. So after reviewing glucose uptake in the major body tissues, it's time to answer the question, how does glucose uptake in normal tissue compare to glucose uptake by the tumor cells? And this answer, is simply shown in the PET scan. Glucose uptake by cancer cells is significantly increased as compared to the normal tissue, which you can see on the PET scan. And it is advantageous because it allows for a fueling of all the metabolic processes that allow these cancer cells to grow and divide at a quicker rate. So, the last question, what modifications may have occurred in cancer cells to alter glucose uptake as predicted above? So I found a study named The Biology of Glucose Metabolization in Cancer Cells by Adewald Fadaka. And they discussed, glycolytic enzymes such as GLUT1, lactate dehydrogenase, and pyruvate kinase as being very highly unregulated in cancer cells, which all of these enzymes are really important in glycolysis. And so this kind of leads to that increase in glycolytic flux for tumor cells, as these enzymes are highly unregulated. And then another important piece of information that I grabbed from the same article is the switch of cancer cells from cellular respiration, which is how, normal body cells do glycolysis, to the very inefficient means of just solely glycolysis for ATP needs. So this switch to aerobic glycolysis is known as the Wahlberg effect, and the reason for this switch is still under speculation. But this means of metabolism is thought to produce metabolic end products to support, its rapid growth, as well as the ability to proliferate under low oxygen tension. So even though this seems, switching from cellular respiration, which makes far more ATP than glycolysis alone, it's just switching to glycolysis, the cells make up for this energy demand by going through glycolysis faster than necessary in normal cells, meaning that these cells need to absorb more glucose in the first place. So by solely using glycolysis as a means of ATP, the cancer cells must adopt a mode of increased glucose import to meet energy demands. This leads to the expression of GLUT1, GLUT3 glucose transporters, as discussed before, that overexpression of the transporters, and an overexpression of those glycolytic enzymes that I talked about before. So that's it for case 2, and then case 3. Case 3 discusses insulin in a different way, giving us serum insulin levels at baseline and then an hour after our glucose load. And this is for a 5-year-old boy who's brought in to a doctor for his annual visit. And this case kind of wants us to put information together about the symptoms that he's reporting to predict what is going on in the biochemical sense. So the main symptoms that are important in this case are drowsiness, persistent thirst, and frequent urination. After hearing these symptoms, my first thought goes to hyperglycemia, because these are common symptoms of individuals with continuously high glucose levels, and hyperglycemia is often found in diabetic patients. So this diagnosis would also make sense based on the individual's serum insulin levels. At baseline, the patient's insulin level is 0.8 milliunits per liter as compared to the control, which is much higher at, around 6 plus or minus 2. And even following a glucose load, the patient's insulin levels are far below the control, showing 5 milliunits per liter as compared to the control, which is 40 plus or minus 11 milliunits per liter. And that is a major difference to take note of. And these low levels of insulin in a patient his age normally arise from your type 1 diabetes, which is when the beta cells of the pancreas genetically do not produce enough insulin because these cells are being attacked by the immune system, and so without, a proper insulin levels, glucose will accumulate in the blood because cells do not have the hormonal signal to take up glucose. So the next question to touch on asks us, what would you predict the patient's fasted blood glucose levels to be given his current condition? So in this scenario, the boy lacks enough insulin to take glucose out of the blood after a glucose load test. This means that the insulin present will allow for uptake of glucose at a much slower rate following our normal fed state. So you can expect in the fasted state, the patient's glucose levels would still remain elevated above a normal range because the insulin that's produced is not enough, and it cannot effectively signal for cells to take up enough glucose to remove that quick enough from the body before the individual feeds again. So lastly, I want to touch on the regulatory enzymes in this patient's condition. So this patient's condition relates to low amounts of insulin, as we discussed before, which leads to low levels of glucose uptake and then subsequently high levels of glucose in circulation. So at the cellular level, this means that the cell wants to promote glycolysis to produce ATP from the glucose that it can uptake. And it also wants to suppress gluconeogenesis so that more glucose is not released into the blood. So three regulatory enzymes in the production of glycolysis are glucokinase, which is also referred to as hectokinase, phosphofructokinase 1, and then pyruvate kinase. And I expect all of these enzymes to be highly active in this patient's condition to promote glycolysis in order to produce ATP from the glucose that the cells can uptake. So just a review of the enzymes and what they do. So glucokinase is responsible for the conversion of glucose to glucose 6-phosphate in the liver. And especially this enzyme, even though it is relatively similar to hexokinase, it is very active in the presence of high levels of glucose, which is what we see in this patient's condition as they have low levels of insulin. And then the second enzyme I mentioned, PFK1, is responsible for phosphorylating fructose 6-phosphate to fructose 1,6-bisphosphate in glycolysis, which promotes the production of ATP. And then the last enzyme, pyruvate kinase, converts phosphoenol pyruvate to pyruvate that can finally enter the citric acid cycle. So I expect all of these enzymes to be regulated in this individual's condition. So thank you. That is Case Connection 2 for Unit 2. Thank you.